not often thought about.
Right.
How the medicines we use are actually proven to, stay good over time. We've got some great info outlining this, and well, our mission today is to pull out the key stuff.
Yeah, to give a clear, concise understanding of what's quite a complex process, actually.
Exactly. For listeners who want to get that knowledge quickly, thoroughly, this deep dive should do it.
Absolutely.
Mm-hmm.
We're diving into pharmaceutical stability testing, the why and the how,
Mm-hmm.
and honestly, there's some pretty surprising aspects to it.
Okay, I'm ready. Let's unpack this. So the materials suggest there's sort of a twofold purpose here for stability testing. What are we looking at?
Well, yeah, fundamentally two main goals.
Yeah.
The first big one is generating solid evidence, like real proof of how a drug's quality changes over time.
Quality changes.
Yeah.
Due to what exactly?
Due to environmental factors, things like temperature, humidity, even light exposure. It's about scientifically watching and documenting those effects.
Okay, that makes sense. And the second purpose?
The second, and just as critical, is using all that data to set, well, two things. The retest period for the drug substance itself, the raw ingredient,
Right.
and the actual shelf life for the finished product, the medicine you buy. And this directly tells us the expiration date and how you should store it.
Ah, okay. So those expiration dates aren't just plucked out of thin air. They come from this testing.
Precisely. They're rooted in careful scientific observation.
Got it. Now, the sources talk about different types of stability tests. Let's start with the one that sounds most straightforward. Real-time studies, or shelf life studies.
Yeah, real-time studies are, well, pretty much what they sound like. You take the product in its final packaging and store it under the recommended conditions.
Like normal room temperature.
Exactly. Normal temperature, normal humidity, whatever is recommended. And you keep it there for its entire intended shelf life.
Which could be.
Oh, often quite long. Maybe
Mhm.
12 months, sometimes 24, even 36 months or more for some products.
Yes.
The gold standard, gives the most accurate picture long term. But obviously it takes time.
Right, a lot of time. So during those potentially years, what are scientists actually looking at? What are they measuring?
They're tracking a whole range of things really meticulously. First, there's the appearance, any color changes, physical deterioration, that sort of thing.
Okay.
Then physical attributes like how quickly a tablet dissolves, the consistency of a cream, maybe particle size in a suspension.
Mm-hmm.
Then the chemical side, super important. Is the potency still there? Are there any degradation products forming? What's the pH doing in a liquid?
Degradation products will definitely need to come back to those.
We will. And finally, microbiological aspects, checking for contamination and making sure any preservatives are still working properly over time.
Okay, quite comprehensive. How often are they doing these checks during, say, a 2-year study?
There's a set schedule.
Mhm.
Typically, you test right at the start. That's time 0.
Mm.
Then maybe 3 months, 6 months, 9, 12 months. Then usually annually after that, up to the end of the proposed shelf life. These checkpoints build up that timeline of quality.
Makes sense. But as you said, waiting potentially 3 years, especially for a new medicine, that seems like a major bottleneck.
It absolutely can be. And that's precisely why we have accelerated stability studies.
Ah, the faster ones. How do they work?
So accelerated studies deliberately speed things up. Instead of nice normal conditions, you expose the product to harsher conditions.
Harsher how?
Think higher temperature and higher humidity. A common condition mentioned is 40 degrees Celsius and 75% relative humidity.
Okay, so definitely warmer and more humid than usual.
Exactly. And you do this for a shorter time, maybe 3 months, often 6 months, sometimes up to 12.
And the goal here is?
The goal is really twofold. First, to stress the product, to sort of force degradation to happen faster. This helps identify potential weak points and break down pathways much quicker than real-time studies.
I see, like an early warning system.
Precisely. And second, the data can be used, often with statistical modeling, to help predict the long-term shelf life more quickly. It also really informs how robust the formulation is and confirms the right storage conditions.
Okay, so real time for confirmation, accelerated for prediction and speed. What about pushing things even further? I think the sources mentioned forced degradation, stress testing.
Yes, exactly. Forced degradation or stress testing takes it another step further.
Mhm.
Here, you're intentionally subjecting the product to really extreme conditions,
Extreme light.
like very high heat, maybe intense light, extreme humidity, or even different pH conditions, way beyond anything you would normally see. You're actively trying to make it degrade.
Okay, why would you want to break down the drug intentionally?
It sounds counterintuitive, right? But it's critical for a couple of reasons. First, you want to identify all possible ways the drug could break down. What are all the potential degradation products that might form under any stress?
Ah, okay. Mapping out the worst case scenarios.
Exactly. And second, this is absolutely vital. It helps you develop and, crucially, validate your analytical testing methods.
The methods used to check the quality.
Yes. You need to prove that your test methods are stability indicating,
Stability indicating, meaning
meaning the test method can accurately measure the drug itself and separate it from, detect, and quantify any potential degradation products. If you haven't forced degradation to happen, how do you know your test can even see those breakdown products if they do form later?
right. You need to know your detector actually works for the things you're worried about.
Precisely. Forced degradation helps prove your tests are fit for purpose.
Okay, this is starting to build a picture of a very rigorous process. It sounds like setting up one of these stability programs needs a lot of planning.
Oh, absolutely. You can't just, put some samples on a shelf. It all starts with a highly detailed stability protocol. That's the blueprint.
The blueprint, what goes into that? What kind of details are we talking about?
Well, pretty much everything. It starts with detailed product information, the exact formulation, the batch numbers you're testing, and critically, the packaging it's going to be in.
Okay.
Then it specifies the exact testing conditions, temperature, humidity, light, tolerances for those conditions.
Yeah.
Very precise.
Right.
It lists all the test methods you'll use for each attribute, potency, dissolution, appearance, degradants, and proof that these methods are validated.
The stability indicating methods we just talked about.
Exactly.
Yeah.
Then the sampling schedule, those time points like 0, 3, 6 months, etc. And crucially, the acceptance criteria.
Acceptance criteria.
Yeah, the predefined limits or specifications the product has to meet at each test point to still be considered acceptable quality.
Okay, so pass-fail benchmarks.
Essentially, yes.
Yeah.
And finally, the protocol includes plans for data analysis, how you'll handle the results, any statistical methods, and how you'll ultimately use the data to propose that retest period or shelf life.
Wow, that is a lot of detail. And potentially a huge number of tests if you have different strengths of a drug or slightly different formulations or container sizes.
It can be, absolutely, which is why the industry uses some clever strategies to optimize the testing load, to make it more manageable without sacrificing data quality.
Okay, like what?
Two key techniques mentioned are bracketing and matrixing.
Bracketing and matrixing. Let's start with bracketing.
Okay, so bracketing is where you might test, say, only the highest and lowest strengths of a tablet formulation.
Instead of testing the middle strength too.
Right. The idea or the assumption is that the stability of the intermediate strength will be represented by the stability at the extremes. You might also bracket container sizes, testing the smallest and largest.
Ah, okay, so it reduces the number of samples you need to test at each time point.
Exactly. It leverages the idea that stability is often related to these variables in a predictable way.
That makes intuitive sense. What about matrixing then?
Matrixing is a bit more complex statistically, but the concept is similar, reducing the total testing burden. Instead of testing every single combination of, say, batch, strength, packaging type, and testing condition at every time point.
Which sounds like it could explode into a massive number of tests.
It really could. So with matrixing, you test a carefully selected fraction of all those possible combinations at each specific time point.
So like at 3 months, you test these specific combinations, and at 6 months, you test a different subset.
Kind of, yeah. The design ensures that over the course of the study, you still get enough data on all the factors, but you don't test every single possibility at every single date. It requires careful statistical design, but it can significantly cut down the number of analyses needed.
Interesting. So smart ways to be efficient. Now, something you mentioned briefly there, and it really jumped out from the source material, was the packaging, the container closure system. Why is that so critical?
Oh, it's absolutely fundamental. Stability testing must be done on the drug product packaged in the container closure system that's intended for marketing.
The exact bottle, or blister pack, or vial.
Exactly. Including any secondary packaging like the carton, sometimes even the label. Because the packaging itself isn't inert, it can interact with the drug.
Interact how? I mean, it's just a container, right?
Well, you might think so, but things can happen. Components from the plastic bottle or the cap liner could potentially leach into the drug product over time.
Oh, okay. Contaminating it.
Potentially, yes. Or, conversely, the drug itself might absorb or bind onto the surface of the container.
Meaning less drug is available.
Exactly, reducing the effective dose. Or the container might not protect the drug adequately from moisture or light, even if the drug itself is stable in isolation. So the packaging is an integral part of the product's stability profile.
Wow, okay, so even the bottle the pills come in is a huge consideration. I wouldn't have necessarily thought about leaching or binding like that.
It's a key part of the assessment, definitely.
Right. That leads us back nicely to something we touched on earlier, these degradation products. You said identifying them as a major goal. Why are they such a big deal?
They're a huge focus because, well, firstly, a degradation product is by definition not the intended drug. And these new chemical entities could potentially be harmful. They might be toxic,
Okay, that's obviously bad.
very bad.
Mm.
Or, even if not toxic, they might simply be inactive. But if the active drug is breaking down into inactive stuff, the medicine loses its effectiveness. The patient isn't getting the therapy they need.
Right, so either toxic or ineffective, neither is good. Where do these degradants actually come from?
Several potential sources. You can get interactions between the main active drug and the other ingredients in the tablet, or liquid, the fillers, binders, what we call excipients.
Ah, the non-drug stuff.
Yep. Or, as we just discussed, interactions with the packaging leaching can introduce things that react, or the drug reacting with the container surface.
Okay.
And then there's just the inherent chemical instability of the drug molecule itself. Over time, exposure to heat, light, moisture, or the wrong pH can just cause the molecule to break down on its own.
So it seems like there are multiple avenues for these unwanted things to appear.
There are, which is why finding them and controlling them is paramount.
And that brings us back to those stability indicating test methods you mentioned. You said chromatography, like HPLC, is key. How does that work to find these specific things?
Right. High-performance liquid chromatography, HPLC, is really the workhorse here. Think of it as a very sophisticated separation technique,
Separating what from what?
separating all the different components in the sample. You inject a tiny amount of the dissolved drug product into the HPLC system. It travels through a column packed with special material, and different molecules travel through at different speeds based on their chemical properties, size, polarity, things like that.
Okay, so it sorts the molecules out.
Exactly. Ideally, the active drug comes out at one specific time, and any degradation products come out at different times, separated from the main drug and from each other.
And then how do you know what they are and how much is there?
As the separated components come out of the column, they pass through a detector. Often it's an ultraviolet or UV detector. Many drug molecules and their potential degradants absorb UV light. Right. The detector measures how much light is absorbed at specific wavelengths. The time it takes for a compound to come out helps identify it, and the size of the absorption peak tells you how much of it is there.
And these methods have to be sensitive enough to pick up really small amounts.
Oh, absolutely. Often, the limits for degradation products are very low, maybe less than 0.1% or 0.2% of the active drug level. So the method needs to be sensitive and accurate down to those trace levels.
And just to connect back, you said the forced degradation studies are used to prove these HPLC methods work.
Yes, that's a crucial link. When you do forced degradation, you generate a whole mess of breakdown products, hopefully representing everything that could form.
The worst case scenario stuff.
Right. Then you run those stress samples through your HPLC method. You need to demonstrate that the method can successfully separate the main drug peak from all these new peaks generated by the stress.
To prove it can see the difference.
Exactly. If your method shows a single, clean peak for the unstressed drug, but then shows multiple well-separated peaks for the stress drug, including the diminished main peak, that gives you confidence the method is truly stability indicating. It can track the drug going down and the degradants going up. And you need to do this validation for both the drug substance alone and the final product in its packaging.
Got it. That validation step sounds critical. And you also mentioned earlier that the accelerated studies, the faster ones, are good for finding these degradants early.
Yes, that's another major benefit of accelerated testing. Because you're pushing the degradation faster, you're likely to see potential degradants show up in, say, a 6-month accelerated study that might only appear after 2 years in real-time conditions.
So it gives developers a heads up.
A really valuable heads up. It allows formulation scientists to perhaps tweak the formulation, maybe add a stabilizer, or work with the packaging engineers to choose a more protective container, all much earlier in the development process. It helps build quality and stability in from the start.
This whole process is, well, it's incredibly detailed and interconnected, isn't it?
It really is. A lot of careful science and planning.
How drugs are essentially put through these sort of stress tests, and why, even with all the sophisticated predictions out there, real-world results are still, well, king.
That's a great way to put it. Still the ultimate proof.
Okay, let's unpack this then. Starting with the idea of how drugs actually break down. One thing that really stood out from the material was this concept of a forced degradation profile. What exactly is that, and why should we care?
Well, okay. So to figure out how a drug might naturally degrade over its time on the shelf, scientists first have to intentionally push it to the extreme.
Oh.
So a forced degradation profile is like a catalog of all the potential ways a drug could break down, but under really harsh conditions.
Mhm.
Think super high heat, maybe intense light or exposure to strong chemicals, that kind of thing.
Wow.
By doing this, researchers can identify all the possible breakdown products,
Yeah.
the degradants that might, theoretically form later on.
So they're deliberately trying to break it.
That's the gist of it, yeah. You deliberately try to break the drug to understand all the ways it could potentially go wrong down the line. It's sort of like quality control on steroids, making sure your medicine stays as it should.
Okay, that makes sense. And the document you shared had a really helpful picture for this, a chromatogram. It showed thermally degraded indomethacin. Now, for those of us who aren't lab scientists, can you sort of walk us through what we're actually seeing there?
Sure. Think of chromatography as a technique to separate different molecules in a mixture. In this picture, the main active ingredient, the indomethacin, it shows up as that big peak. It's labeled peak 5, and it appears at about 4.8 minutes on the readout.
Okay, the main drug.
Right. But what's really interesting are the other peaks that pop up after the drug has been deliberately heated. You see peak 1 showing up earlier at 3.8 minutes, and peak 2 even earlier at 2.9 minutes.
And those are the breakdown products.
Exactly. Those are the breakdown products that the degradants form because of the heat stress.
Yeah. So the fact that these different molecules show up at different times, that separation, that's the really key thing here, isn't it?
Precisely. That separation tells us that the analytical method being used in this case is something called high-pressure liquid chromatography, or HPLC.
Hmm.
It can clearly tell the difference between the original intact drug and any bits of it that have broken down.
Ah, I see.
And that's why the text mentions this method can be called stability indicating.
Okay, stability indicating assay. So it's basically a test that can tell us if the drug is still, in good shape or if it's starting to fall apart.
That's it.
And the document points out peak 2 as the main thing it breaks down into, and peak 1 is sort of a secondary breakdown product. Does it matter which things appear and how much?
Oh, absolutely. It matters a lot. Knowing which degradants are the main ones and in what amounts they form helps scientists understand the primary pathways of degradation, the main ways the drug breaks down. And this knowledge is super important. It can lead to changes in how the drug is maybe formulated or even manufactured to make it more stable in the first place.
Okay.
It also helps in creating specific tests to look for and measure these particular breakdown products during the actual stability studies later on. And crucially, it helps regulators set safe limits for these impurities to protect patients. You can't just have unlimited amounts of breakdown stuff in there.
No, definitely not. Okay, so that leads us nicely to the idea of trying to, well, predict how quickly a drug might decompose. The information you shared mentioned using kinetic models. What are these, and I guess why aren't they the only thing we need to rely on to figure out a shelf life?
Right, so the idea behind kinetic models is that you can use mathematics, essentially chemical reaction rate theories, to predict how fast a chemical reaction will happen. In our case, that reaction is the drug breaking down. And it's often based on factors like temperature. So in theory, if you know how quickly a drug degrades at, say, a high temperature in the lab, you could use a model like the Arrhenius equation, perhaps, to estimate how quickly it will degrade at a lower normal storage temperature over a much longer time.
Sounds like a handy shortcut. Saves time, right? So what's the catch? Why isn't it that simple?
Well, the big problem, as your source correctly points out, is that these models often work best in a sort of perfect, simplified world, like just the drug molecule itself, in a simple solution. They might not fully take into account the complex reality of a finished drug product, the pill or the injection you actually use. Things like all the inactive ingredients, what scientists call the matrix.
The fillers and binders and stuff.
Exactly, all that other stuff. Or if there are other active drugs in the same product. And really importantly, physical stability issues. Things like how well the tablet dissolves over time, or if the drug starts forming tiny particles or crystals. These physical changes are often not fully captured by simple chemical kinetic models alone.
That's a great analogy you used earlier, like predicting a car engine's life just by looking at metal corrosion in a lab without considering the whole system and driving stress.
Yeah, something like that. Yeah. You miss the interactions, the physical stresses, the complexity of the final product and its packaging.
And this is why the document really stresses that there's ultimately no substitute for determining the shelf life empirically. Meaning you have to get real-world data on the actual finished product in its final packaging.
Exactly. You have to actually see how it behaves over time under conditions that mimic or are the actual intended storage conditions.
So, okay, we've seen how scientists intentionally break down drugs to understand their weaknesses using forced degradation. Now, let's talk about how they use that information and these stability indicating tests to actually determine how long a drug will stay good on your shelf. This brings us to the different types of stability studies mentioned, real-time studies and accelerated studies. Let's start with real-time, the ones that give us that crucial empirical real-world data. What are the main things to know about these?
Okay, real-time stability studies. These are really the bedrock, the foundation for figuring out a drug product's true shelf life. As the name implies, these studies happen over the entire time the product is intended to last. So if it's a 2-year shelf life, the study runs for 2 years.
Wow, okay. Long-term.
And critically, it's done under storage conditions that are the same as the maximum recommended on the product label. And very importantly, the drug product being tested has to be in its final primary packaging, the actual bottle or blister pack or vial it'll be sold in.
So not just the drug substance on its own.
Definitely not. It has to be the finished product in its market container. The whole goal here is to get solid, direct proof of its stability over its entire proposed lifespan under normal storage.
And what are some of the specific rules or requirements for these real-time studies? The document you shared seemed to have quite a detailed list.
Yes, there are several pretty strict requirements to make sure the data is robust and reliable. First off, the way the drug is formulated and manufactured has to be exactly as described in the regulatory filings, the registered details.
Makes sense.
Second, the product absolutely must stay in its registered and closed container throughout the entire testing period. You can't just leave it open on a bench.
Right.
Then there's the number of batches. Initially, for registration, you generally need data from at least 3 primary stability batches, usually production scale or pilot scale that mimics production.
3 different batches. Why 3?
Good question. It's about consistency. You want to show that the stability isn't just a fluke of one particular manufacturing run, but that it's reproducible. It gives confidence that most batches will behave similarly.
Okay, so consistency.
Exactly. And then after approval, there's usually an ongoing commitment to put at least 1 production batch per year onto a stability program,
To keep checking.
to keep confirming that nothing has changed in the manufacturing process that might affect stability down the line. Also, if the initial stability data came from a smaller pilot scale batch, you still need to test the first 3 full-scale production batches.
Right. And what about different strengths, like a 10 milligram and a 20 milligram tablet?
Yeah, the guidelines cover that too.
Yeah.
For drugs that come in different strengths, you generally need to test at least 3 batches of both the lowest and the highest strengths. You might be able to bracket or matrix the intermediate strength sometimes, but the extremes are usually tested.
Okay. And the conditions, temperature and humidity.
Yes, specific conditions are mandated. For long-term, real-time studies aiming for typical room temperature storage, it's usually 25 degrees Celsius, plus or minus 2 degrees.
So quite tightly controlled.
Very tightly controlled. And 60% relative humidity, plus or minus 5%, if humidity is relevant for the product, which it often is for solid pills, for instance. And this goes on for the minimum duration of the proposed shelf life, often 12 months initially, then extending longer.
That sounds incredibly rigorous, really thorough.
It has to be. Patient safety depends on it.
Absolutely. Now let's shift gears to the other type mentioned. Accelerated condition studies. These happen much faster, right? What's their main purpose compared to the real-time ones?
Exactly. Accelerated studies are designed, well, to accelerate the degradation process. They do this by exposing the drug product to much tougher conditions, significantly higher temperatures, and often higher humidity, too.
Like putting it in an oven, almost.
Sort of, yeah. A very controlled, specific kind of oven. And these studies run over a much shorter period, usually somewhere between 3 and, say, 12 months, often just 6 months for initial registration data.
And the goal isn't to set the final shelf life here.
Not directly, no. The main goal is to get an early prediction of the shelf life, and importantly, to better understand how the drug breaks down under stress. This is especially useful during the early stages of product development, and when you're first applying to get the product approved or registered.
So it's like a fast-forward button on the aging process of the drug to see potential problems sooner.
That's a perfect way to think about it. By seeing how the drug behaves under this heightened stress, scientists can get an early indication of its likely long-term stability profile. It helps identify potential weaknesses quickly.
which could help get needed drugs to market faster, potentially, if they look stable under stress.
Potentially, yes. It supports the proposed shelf life based on real-time data, and it can help justify excursions outside labeled storage conditions sometimes. It also helps define the packaging requirements, like does it need extra protection from moisture?
Okay. And what are the key requirements for these accelerated studies? Are they as strict in terms of batches and things like that?
They still have important requirements, but slightly different focus. You absolutely still need to use a validated stability indicating analytical method that's crucial.
Right, the test that can spot the breakdown.
Yes. However, because the main focus here isn't necessarily proving batch-to-batch consistency in the same way as real-time studies for shelf life setting, often data from just one batch of the product is considered sufficient for an accelerated study during development or registration.
Ah, okay. Fewer batches needed.
Generally, yes, for the core accelerated study. And the typical conditions are pretty standard. A temperature of 40 degrees Celsius, plus or minus 2 degrees.
Flat warm?
Oh, yeah. And critically, that 40 degrees C has to be at least 10 degrees higher than the long-term storage temperature. So if long-term is 25 degrees C, 40 degrees is more than 10 degrees higher. And usually 75% relative humidity, plus or minus 5%. Again, if humidity is relevant, for a minimum duration, often 6 months.
Okay, 40 degrees and 75% humidity for 6 months. That sounds pretty intense.
It is intended to be stressful for the product to force degradation to happen much faster than it would at room temperature.
The document also mentions something called intermediate condition testing. When would that come into play?
That's a good point. Intermediate testing is kind of a fallback or an additional step. It's typically triggered if you see significant changes happening during the accelerated testing at 40 degrees, 75% RH.
Ah, so if the drug starts breaking down too quickly under the accelerated conditions.
Exactly. If it fails specifications or shows significant degradation early on at 40-75, then testing might be done at an intermediate condition. This is usually something like 30 degrees Celsius and maybe 65% or 75% relative humidity.
And why do that?
It helps get a clearer picture of the degradation rate under conditions that are still stressful but less extreme than the 40-75. It can help in establishing appropriate storage conditions or perhaps justifying a shorter shelf life if the product isn't stable enough for the initially desired term.
Okay, that makes sense. And speaking of significant changes, you just mentioned that term. The information you provided gave a really clear list of what actually counts as a significant change during stability testing. Could you walk us through some of those key signs or red flags?
Absolutely. These are the specific triggers that scientists and regulators watch out for very closely during stability studies. They signal that the product's quality might be compromised.
Okay, what's on the list?
Well, first, a loss of potency. Generally, a 5% or more drop in the amount of the active drug compared to its initial measured value is considered significant.
So if it starts out at 100 milligrams and drops below 95 milligram?
Precisely.
Yeah.
Another big one is any degradation product reaching or exceeding its preset specification limit. These limits are based on safety and qualification studies.
Right, those breakdown peaks we saw in the chromatogram earlier.
Exactly. If any of those get too high, that's a significant change. Then there are physical and chemical properties. The product exceeding its allowed pH limits, if it's a liquid or semi-solid.
Okay.
Also, for solid forms like tablets or capsules, failing to meet the dissolution specification, meaning it doesn't dissolve properly anymore, which could affect how the body absorbs it.
That's critical.
Very critical. And then there are more observational things and significant deterioration in the appearance, like color changes, phase separation, precipitation, cracking, or other physical attributes. Basically, if it stops looking or behaving like it should.
So appearance matters too.
It definitely does. And finally, the guidelines, like the ICH guidelines mentioned in the source, state that failing to meet specifications under those accelerated conditions we discussed, 40 degrees C 75% RH, or sometimes intermediate conditions like 30 degrees C 75% RH, depending on the climate zone the drug is intended for, within a certain time frame, often 6 months, is itself defined as a significant change.
That's a really helpful and quite concrete list. It really highlights just how many different things are being meticulously checked to make sure a medication stays within acceptable quality standards throughout its life.
Exactly. It's a really comprehensive evaluation. It's definitely not just about checking if the main ingredient is still there. It looks at the overall integrity and performance of the entire drug product.
So to sort of wrap things up for our listeners, you should now have a much clearer picture, hopefully, of the really rigorous testing that pharmaceutical products go through, all to ensure they remain safe and effective, over time. We've looked at how scientists analyze the potential breakdown of medications using things like force degradation and stability indicating tests.